Today in this tutorial we are going to learn about Python skills, but this Python programming tutorial will be only for the bioinformaticians out there or those who are doing genomics research. So we are going to do a mini project as well, and also we are going to learn how we can set up Anaconda package manager, how we can initiate conda environment, and also we can learn about how to launch Jupyter Notebook to write Python codes. And then we are going to do a mini project on Gene Expression Omnibus, which is a popular database of RNA sequencing files. And also we are going to write Python code to download Gene Expression Omnibus datasets. And I'm going to explain you line by line so that you can understand and implement it. So the first thing that we require is Anaconda package, and this is the package manager for installing different kinds of tools, not only Python but also R packages as well. And this is one of the best package manager for bioinformatics. Okay, the next thing that we are going to learn is how to launch Jupyter Notebook from the conda environment, which is IDE for Python. And the next thing that we are going to do is we are going to start a mini project to write Python code to download Gene Expression Omnibus datasets from NCBI. So yeah, let's begin. The first thing that you want to do is visit this Anaconda site. I'll put the link in the description below, and download the package from Anaconda. It depends on your operating system. If you are using Windows, then Windows package will be automatically downloaded. Since I'm using Mac, so the package manager, I have already downloaded the package manager. So let me just begin to install it. So this will be the setup window, and you press continue, 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 agree, and then install for me only. Okay, and then we go for installation. So while writing the files, I'm also going to show you the tutorial on Anaconda website itself. So here you can see that how you can install on macOS. I think they have also description for Windows. If you search this website from your Windows operating system or Linux operating system, you will find the tutorial for Windows as well. So you can also install it from command line, but this is much easier to install from the package manager itself. So I think it will take some time. It is less than a minute. Okay, then we wait. And the project that we are going to do, as I mentioned previously, is from we go to the Gene Expression Omnibus, and then we download a particular data set from this Gene Expression Omnibus. So what we are going to download is the metadata and also the raw counts file, which is required to process for DESeq2 package or RNA sequencing, right? So this is a small data sets. This is just for this tutorial purpose, but also you can download big files as well from Gene Expression Omnibus. So I'll put the so we are using this lung cancer data sets from Gene Expression Omnibus. This is related to Homo sapiens, and you can read about the description when you try to initiate a project in RNA sequencing. Then you can read the metadata from here. Also you can download from by clicking into the link, but I just want to focus on Python programming skills in this tutorial so that you can learn a little bit of coding and you can showcase your skills rather than depending on going into the browser and then downloading by clicking these type of links. You can click here and it will be downloaded. I mean, that's easy, but how you can achieve it by using Python programming, that's really important, so that you can learn Python alongside and also you can implement your skills. Okay, the installation is complete, and you can see this pop-up window as well, and you can close this. Okay, now you can see that Anaconda Navigator is automatically launched. This is the graphical user interface for Anaconda Package Manager. So from here you can launch Jupyter Lab, notebooks, even VS Code, AWS, and many more if you want to write specific Python programming integrated development environment, or we call it as IDE. Then you can use PyCharm as well, and also you can install RStudio from Anaconda itself. So it's a really good package manager for software people, for IT people, and especially for bioinformaticians. So it's in the home directory, and if you go to the environments, you can see that a base environment is automatically installed. And I'll also tell you why, what is the importance of this base, and also you can learn many tutorials. If you want to learn Python, here you can find many tutorials for R, PyCharm. You know, there is no limit in learning skills. So Python is one of the best thing that I would say has been developed by, you know, whoever it is. So he should get a Nobel Prize or something. And then you have the community where you can reach out to experts and ask questions, and also get your queries solved over there, like Stack Overflow for any IT related queries. So you go to the Stack Overflow. Anaconda also have different types of communities, right? Okay, so those who don't want to go for command line installation, they can use this graphical user interface to install different packages in here. So if you go here and then click all, and you can search for packages from here. So if you want to search a package like BioPython, you can see BioPython. If you click this, it will automatically be installed. So here are the purpose, but we are not going to do that. We are going to do this from the command line terminal. Okay, so let me close this, and let me tell you why this from where this base thing comes up. Okay, so if you open your terminal, you can see that it's automatically, the base environment of the Conda has been activated automatically. So how this is done? So if you open your bash RC or ZSH RC, those who are using different types of shells. So I'm using ZSH RC, and you can see that this particular chunk of code has been written automatically if you install Conda package or Anaconda. Okay, so this is automatically being written to your shell file. That's why whenever you open the terminal, you get the base environment. Okay, so let me close this, and now we are going to open the tutorial for the Conda environment. This is the user guide for Conda, and this is the latest version. You can go through this and learn how to install it for Windows as well. Here you can see. Okay, so we are going to launch the terminal, and also we follow the tutorial alongside. So the first command that they are telling is, if you write Conda, since your base environment is activated, your executable file Conda is already there in the path, right? So Conda --version, and you can see the latest version, that is 23.7.4, has been installed over here. Okay, and if you search for then also you can update the conda if you don't have the latest version, or those who have installed already conda on your operating system, then you can update conda, right? So it will update the conda packages. It will take some time, but yeah, it will update. Since I have recently installed, so that's why I don't think so it is going to ask me to update my package, but yeah, let's see. Okay, I'll put the link in the description so that you can have a look how to initiate environment and how to read the bash rc or datasets rc file, so that you can know that why your base environment is activated. Let's see. Oh yeah, you can see that different types of, so if I write conda update conda, you can see that these packages, the following packages will be downloaded. Okay, because each and every package has different build versions or different types of versions, so that's why it gets updated all the time. And you can see also the size of the files that are going to be downloaded, that is 237 megabytes. And the following new packages will be installed. These are the new packages that are going to be installed, and these packages will be removed because new packages will be installed. So it's done automatically, you don't have to worry about anything. And all these packages that are pre-installed while installing the Anaconda, these are going to be updated. Okay, so you proceed with yes, and you can see this matrix, like, you know, movie that will show the progress of the download, and it will be soon completed because the file size is not so big. Right, let me just make it big. Yeah. There you can see. Now we are also going to learn about how to manage the environments. Like in my previous tutorial, you have seen that we have activated Quarto package, where our environment has automatically invoked, and whatever the packages that you have installed inside that R code has been locked. And if you want to transfer that to your colleague, or somebody else wants to use what you have done previously on that specific project, they can activate the R environment, and they can also get the results similar to what you have obtained in your project. Okay, similarly, the conda environment is another way of managing your environments. The packages that you are going to install will be, the version of the packages will be there forever. Okay, unless you update it. So yeah, so after the completion, it will always say that preparing transaction done, verifying the transaction done, and executing the transaction that is done. So it will be done soon. Yeah, this is one of the most important command that you always encounter, and this will help you to manage different environments. Okay, so I copy the code. Let's see if it's complete. Yeah, now it's complete. Okay, so if you type conda info envs, you can see that how many environments are there. Oh, sorry, the other way around. Conda info envs. You can see that you have only one environment, that is your base environment. And in the base environment is kind of a root folder for your Anaconda, where each and every packages have been installed. Okay, But if you start a mini project, or if you start your project on RNA sequencing per se, and you want to install BioPython, so you want to manage that version in another environment so that your base environment is not affected, right? So it's really important to manage different kinds of environments per project so that your base environment is not affected at all. So what we are going to do is, now we can create another environment where we are going to do this mini project on Gene Expression Omnibus. Okay, and since we are going to use Python, so let me just copy this. So the command would be conda create and hyphen name. You can name anything. Since we are going to do the mini project on Gene Expression Omnibus, so I write GEO, and this is the version of the Python that you are going to use in this particular environment. So if you click enter, so it will create another environment called Gene Expression Omnibus. And since you are using Python 3.9 over here, you can install any version of Python or any type of package that you need. But since you are dealing with the Python, so I copy the command from the website itself, and you can see that these packages will be downloaded, such as the latest version of the pip, and you can see the Python version, some setup tools, and these following new packages will be installed in your environment. So if you proceed with yes, you can see that the transactions will be done. Okay, and this command will pop up. So you can see that conda activate GEO, that is our environment name, and also you can deactivate this particular environment. So the command to know that what kind of environments have been there, so what you will find is, one is the base environment, that is your root folder kind of thing for Anaconda, and this is the new environment that you have created, that is GEO. Okay, and these files are written to my home directory where Anaconda is there, and you can find a folder called envs, and there your environment is there. So let me clear this out, and you can activate this particular environment by using conda activate GEO, and you can see that your GEO environment has been activated. So now you can do whatever you want. You can install any type of packages that will not affect the base environment. Okay, and this is how you manage different kinds of environments so that everything is written on that particular environment. Okay, so this is how you make. And since you have installed Python 3.9, let's check Python version, and you can see 3.9.118 is installed in Gene Expression Omnibus or GEO environment. Okay. Now if you want to deactivate that particular environment such as GEO, what you can type is conda deactivate. That's it. So if you type this command, the GEO environment has been deactivated and it returns to the base environment. Okay, so now again do conda activate GEO, and it will again activate your environment, right? And one more thing, that if you want to search for some packages that you don't know, you don't want to go into the browser and search for it, but in conda you can also search some packages. So we can also execute this command like conda search, and if you want to install matplotlib, for instance. Matplotlib is one of the package for Python plotting, so you can search for conda search matplotlib, and you can see that matplotlib is available in the conda channel. And you can also install this particular package by installing different channels. So there are mainly 4 types of channels in Anaconda environment. One is conda forge, then Bioconda, R for R packages, and the default one. Okay, so to understand how this channel works, so we are going to add one of the channels that we have just seen. So conda config -- add channels conda forge. If you type this particular channel will be added to the GEO environment, and you can see that, yeah, this channel has been added. Now let's add Bioconda, and you can see Bioconda has been added, and you can type in R as well. So these 3 channels have been added into the GEO environment. Now let's go and search for a matplotlib installation. Okay, so if you go to the documentation of matplotlib, you can install by using pip, or you can install by using conda. So the command would be conda install matplotlib, since it does not require any channels. So it can be installed by using the main channel, but also you can install by using specific channels such as conda forge in this case. So the command would be conda install -c, that means channel, and conda forge, which channel that you are going to invoke, and then your package name. So we are going to copy this command, conda install -c conda forge matplotlib, and you can see that it will it will display the channel information like conda forge, R, Bioconda, and defaults that we recently added, and it can show you the platform as well. And now this will be installed from the channel called conda forge. So it will take some time, but it will be installed. I will also tell you in the base environment, if you want to install matplotlib by using this particular command, let's see what happens. So it will take some time. Yeah, it's done, and you can see that the packages that is required to install the matplotlib will be downloaded, and then you proceed with yes, and you can see this matrix movie where all the packages will be downloaded, and then the transaction has been done. Okay, now since you have added all the channels, now if you search for a specific package such as fastqc or something, let's see in which channels it is available, and then you can specify the channel name, and you can install the package itself. Okay, I think fastqc is available from Bioconda, as you can see from the Bioconda. So the proper way to install fastqc would be add the channel name. So let me just clear this. So conda install -c Bioconda and fastqc. So this is the command to install fastqc by using the channel called Bioconda, and it will be installed. Okay, since I have already installed, you can see that all the requested packages have already been installed. So if you type in fastqc, and then you can see that if it works, then it can pop up the window for that, and the executable file will be executed from that. Okay, and if you want to uninstall any package, if you don't want fastqc like that I have done previously, that I have already installed the fastqc file, fastqc executable. So if you want to uninstall, then the command will be uninstall fastqc, and you can see that it can search for the channels, and it will tell you that these following packages will be removed. So you type in yes, and it will process the transaction. And now if you type in fastqc, it will not show up because you have just uninstalled fastqc. So this is the way that you can manage all your packages. If you want specific package to be in your environment, then you can specify that. Otherwise, you can uninstall those packages. Okay. Now let's try to run Jupyter Notebook from your environment. So you type in the command Jupyter Notebook, and you can see that the command not found will be popped up. It's because the Jupyter Notebook is installed in your base environment. When you open your Anaconda Navigator, and you can see that the base environment has been selected, and your Jupyter Notebook is installed on your base environment. So let me just show you how to. So let me just deactivate this channel, and if you type in Jupyter Notebook from here, you can see that Jupyter Notebook will be start from here. But if you want to access Jupyter Notebook from your particular environment, then Control C, yes, you want to shut down the server. Then let's again activate our environment, and if you want to run Jupyter Notebook from here, the command will be, you can search for Jupyter, and you can type Jupyter, and you can see the channels will be loaded from where you can install Jupyter Notebook. Since you are created this environment, the Jupyter Notebook has not been installed over here. It's in the base environment. So you can see that you can just type in the command called conda install Jupyter, and also you can specify the channel conda forge as well. So let us just install Jupyter and then try to invoke Jupyter Notebook from this particular channel. Meanwhile, let me show you this particular website where there is a conda cheat sheet where you can run different types of commands in your conda in order to look into different perspective of what other available commands that you can explore, and I'll put the link in the description below so that you see install a new package Jupyter Notebook in the active environment. That means you have to install the Jupyter, and then you try to invoke Jupyter Notebook. Let's see. Yeah, now it is going to be installed, and the command to invoke this is Jupyter-Notebook, and also you can type Jupyter Notebook with space as well. So let us try this command. Okay, now it's done. So Jupyter Notebook, and you can see that now the Jupyter Notebook has been installed in your environment, and it will be invoked. You see, now it's there. Your Jupyter environment is invoked from here. So this is how you maintain Anaconda environment. So let me just shut it down. Yes, there are also some other commands that I want to show, that is list, that it will show you what are the packages that has been installed in this particular environment, as you can see. So these are the packages that are installed in this particular environment. And if you go to the base and type in conda list, also the packages which are installed in your base environment will be invoked. Now we can initiate the Jupyter Notebook from the conda environment that we have created, and we are going to perform this Python coding for downloading the Gene Expression Omnibus datasets, which is the raw count file. So if you go to the website, and yeah, I have opened it. So if you go to the website Gene Expression Omnibus and type in some disease, for instance COVID, I'm searching for lung cancer, and we can select a small data set if we like, for instance maybe this one. Yeah, this is selected already. So we can write a Python code to download the raw counts file from Gene Expression Omnibus, this particular database. So what you have to do is, in the same environment, you have to type Jupyter Notebook, or before that, what we can do is we can go to desktop and create a folder called make directory download GEO, and then cd download GEO. Okay, and from here you can initiate the Jupyter Notebook. And you can see that if you look into the folder architecture, there will be nothing, because you have just recently created the folder. So that's why you don't have anything inside the folder. And then you go to the new and create a notebook, and you rename it as something like download geo dataset.ipynb is the extension for the Python notebook. Okay, and if you double click it, it will open the notebook, and it will prompt that whether you are using the Python 3 kernel. Yes, select it. Okay, and then we need to write the Python code in order to download the datasets from Gene Expression Omnibus. So the first thing that you have to do is you have to import different libraries from Python. So one of the library that we are going to use in order to download Gene Expression Omnibus dataset is the request library. So you can start with the hash or the comment. So maybe write import the libraries, import the libraries. So always these comments are necessary, because if you can distribute your Python code to your colleagues or your professor, or maybe you are using it for publication, then these comments will help them to reproduce your results. Okay, so the first thing that we are going to do is import. The import is the function in Python where it can import different types of libraries. So first import that we are going to do is requests, because we are going to request to the Gene Expression Omnibus database to download the file. So let's open the Gene Expression Omnibus database, and let's see which file we are going to download. So if you go to Gene Expression Omnibus, here the keyword, you can type in any type of disease name. So for instance, I'm let's type lung cancer or something, and then search for it, and you can see two types of results will pop up. The first one is the datasets, and the next one is the profiles database. So we are going to download the datasets, that's why we click here, and then we can choose any type of organisms. Here I think we are going to download a small dataset, maybe the first one or something. Let's see. And this is the geo accession number that you are going to download the dataset. Oh, I think there is no counts.file. Maybe we go to the next link, this one. Yeah, here because this is the file that you require in order to process further analysis in bioinformatics, such as RNA sequencing. So you require this raw counts text.file. So find a dataset that provides you the raw counts. So this is the dataset having 4 samples, and I think they have the raw counts file as well. Okay, so this is the dataset that we are going to download. So the first thing that you import the function is the request, because we are requesting the database to download our files. And if you want to know that whether your code works or not, you can click on that particular cell. So this is the first cell that we have written the code, and then you can click here. So this will run the cell which you have selected only. And if there are any errors, it will show up here that the code that you have written has some errors. Okay, so now we don't have any errors, so I think this works. Now we need to define a function. So as I mentioned previously, that we are going to write a function in order to download the dataset from Expression Omnibus. So the comment will go like, define a function to download the dataset. This is a very small example that I'm giving you right now, but you can write many different codes so that you can download the whole library as well. I can provide you the code that I have written for my own purpose, that you can download the metadata as well as all the supplementary files associated with it. I'll show you at the end of the video. So to define a function in Python, the keyword to define a function is def. Okay, it starts with def, then you have to give a function name. For instance, I give download the file. Okay, and then you have to provide 2 parameters. So the first one is the URL that you are going to use, because this is the URL that we are going to use to download the file. And the next thing, the local file name or the file name that you are going to save in your local directory. Okay, and whenever you define a function, it always ends with colon. And if you if I don't put colon there, and if I press enter, it will the next line will come directly under this particular def keyword. And if you try to run this, it will throw an error, because this is a definition function and it has an ending which is the colon. And if you press enter now, it will automatically indent. This is the good function of Jupyter Notebook or any type of code editors that we use. It can indent automatically. Okay, now we have to get the response from the URLs from this particular database. So what we have to define is the response that we are going to get is equal to requests, because this is the library that we are importing, and request get and your URL, and stream is equal to true. I'll tell you what I'm doing. So first let me write this, and if the response dot status underscore code is equal to 200, now you can see that it will indent automatically with open file name wb where to write as file, then colon, then for chunk in response dot iter, the content that we are going to iterate, and then the chunk size will be around 1024. bytes. Okay, and then we file write and the file that we're going to write in this chunk. Else, because this is an if statement, so it always ends with else. Else print that fail to download the file. Fail to download the file. And you can also give the status code response, status code, and the response dot status and code. So this is always if you redirect to any URL, if you want to download some file and it does not download the file, then it will give you a status like 404 error or something like that. Right, then you close the bracket. Now this is the function that you have defined with if statement and else statement. And yeah, let me tell you what I have done exactly. So here I define by using the keyword def the file, the function name that I have defined, and I have provided two parameters. One is the URL that you are going to download the file from, and the file name that you are going to save in your directory. Okay, the next thing, the response and response get URL stream is equal to true. This script makes an HTTP get request to the URL provided. From here we are going to get this HTTP request from the server, and the stream true argument tells that the requests library to download the file in chunks instead of loading it all into memory at once. So it can download the file in chunks. Since here the dataset is very less, so it will not affect while downloading the file. But if you have a big file, then it can convert into chunks and then it gets downloaded, so that you don't have any memory leak or memory error. And this particular code, if response status code is equal to 200, this line checks if the HTTP request was successful or not. A status code of 200 means okay, indicating that the request was successful and the server responded with the data requested. And now the next line of the code. So this with open, this line opens a file with the name provided in the file name, and the WB mode means write the binary, the write binary mode. Okay, and as the file being downloaded is compressed as ggip text file, so whatever you are downloading, it's in ggip compress file, so that's why it downloads as a binary file. Okay, and the next code for chunk in response. So this line iterates over the content of the response in chunks, each size of 1024 bytes. Iterating in chunks is memory efficient. I mean, this is the way that you can download the file from the server. If it is a big file, then it can divide into chunks. In this case, it's 1024 bytes. Okay, and it can be applied for large files or big files that you have in different databases. Okay, and the next thing, that file write chunk. So inside the loop, each chunk of the data is written to the file, and this process continues until all the chunks of the file have been downloaded and written to the local file. And the else statement is, if this is successful, then this will not be printed. If this is unsuccessful, this will be printed with the status code. Okay, so now the function has been defined. We can Now try to run it. Let's see if it works. Yeah, it works because it does not throw any type of error. I'll tell you why, if any error comes, how you can rectify the error. If you get an error or you have messed up with the code or something, I can tell you about that as well. So now you can actually provide the URL. So you define as URL and your file URL from here. So you just copy this link and then provide with double quotes. Okay, so this is the URL, and you can define the file name, because this is the function that you define here. So there are two parameters, one is the URL and the next one is your file name. So you provide the URL and now you provide the file name. For instance, I can tell the counts dot text dot cz, something like that. Okay, now you call the function. So download, so the function name that you have defined. So now you can call the function. So first you define the parameters. So I can write it over here maybe. First define the parameters inside the function, and then here you can call the function. So download file, this is your function name over here that you have defined. So download file and the URL that you have provided over here, comma the file name. Yeah, this is it. I mean, this is a small Python code in order to download the dataset from the Gene Expression Omnibus. So let's try if we can run this. You can see true is not defined. I did a mistake over here. So the stream true, it should be in small letter, not in capital letter. Now see it turns into green. So yeah, this was the mistake. So and also again, this is a good practice that we get some errors over here, and you can see that the errors, if you encounter any error, so it will be popped up here with this orange bar, and you can see that okay, this is not correct or something is wrong. So now we have rectified this, because I defined it in capital letters, it has to be in small letters. So now if we click this, let's see, yeah, it works. So I delete this. Okay, it was not the error was something else. So I paste it over here. Now we run the code again and see. Yeah, now you see, now it works perfectly. Now if you go to the folder, or maybe go to the home folder, and if you, I think, yeah, now you can see that your file has been downloaded. If you click this, you can see, okay, you cannot open this because this is count.text.gz. So in here you have actually downloaded the dataset from the Gene Expression Omnibus. So we can try with other option also, which provides the, yeah, you can see here, you can copy this link and then go here, replace this, and you can also define the accession number and the count file. So I define the accession number with the file name underscore, and then we try to download this. Let's see if it down. Yeah, now you can see it is downloaded. So if you reload this, you can see that your file has been downloaded. So this is how you write the code. So I have also written a code that can actually download data sets Okay, let me open this. So I have used a function called GEOparse. So this is the function that I have used, and the FTP library function I have used, and the OS import libraries, the libraries I have used, and have defined the FTP library function. I have defined the geo data, and actually I print everything from the geo datasets. So actually it can print the title, summary, overall design, etc. So what I have to do is I have to just give the accession number. So let's see if we can download this particular accession number, or we can go back and use this one. And this is the accession number that I have used in that particular code. And you can see that these summary along with the metadata, along with the supplementary files, all will be downloaded in that particular code. So this is the accession number. So if I replace it, this is the accession number that I have used, and I save this. And if I call this from terminal, and the command would be to download the datasets or to access this particular Python code. So we have to go to the directory. This is the directory, and if you ls, this is the Python code, which has an extension of .py. And if you write Python 3, or maybe Python you can use, because you have the default is Python 3, so you can call it as Python and then geo.py. So this is how you can execute the Python script. And if you click here, you can see that, see it can so I can just enlarge it a bit. So what this code does that is that it can check the directory whether it is existing or not, and then it can download all the information about the datasets along with the metadata, and along with the supplementary file it can download from the Gene Expression Omnibus. So one thing that you have to provide is only the accession number, nothing else. Only the accession number you have to provide, and it will download everything from the Gene Expression Omnibus. So I'll upload this code in my GitHub channel, so that you can download it from there, and you can use it to download the datasets from Gene Expression Omnibus. So this is all about Jupyter Notebook, how to handle this, how to write the code. So we have just simply written a simple code to download the dataset by providing the URL, but I can also provide the script so that you can download the datasets from Gene Expression Omnibus without worrying about anything. It can download all the information for you. Now you can see in the folder, you can see that the metadata information has been downloaded, all the metadata information, and you can also see the counts file. So this is the counts file, okay, which will be utilized for RNA sequencing, and also this TPM file as well, which can also be used for RNA sequencing analysis or DE-Sec2 analysis. Okay, so I'll provide this code in my GitHub repository, and until then, see you next time. Thank you very much.